Good morning, good evening, good afternoon, and welcome to this lesson on clinical trials. Now, before we get into it, let's look at some learning objectives. So students who complete this learning module should first be able to define a clinical trial and recognize when clinical trials are used, understand the advantages and disadvantages of clinical trials, know what experimental design features are used in clinical trials, know briefly what the different stages or phases of clinical trials are, know of some of the ethical considerations involved in clinical trials, and to be aware of new emerging approaches to clinical trials. Now let's get into it. So what exactly are clinical trials? Well, clinical trials may be defined as a type of research method that aims to assess and compare the effects, safety, performance, and efficacy of various treatment therapies for humans by performing an experiment involving human patients. Therapies assessed may include medications of either a chemical or biological, such as monoclonal antibodies, nature, medical devices, or biological products containing human tissue or live animal tissues. In clinical trials, participants are divided into at least two groups which receive different treatments, allowing for a comparison of the efficacy of a given therapy to be made. However, in order for useful comparisons to be made, one group of participants must be a control group, for whom the efficacy of a particular therapy is well known. Each group is referred to as a treatment arm. Now, when should clinical trials actually be used? So clinical trials are commonly used in pharmaceutical research, but have not been easily implemented when assessing other medical interventions. For example, clinical trials have been criticized for their use in psychotherapy due to difficulties associated with assessing the effects of long-term and personalized interventions. Clinical trials are also considered inappropriate for the evaluation of surgical techniques due to the recognition that surgeons have different skills, patients have different pathologies, and sham surgery controls cannot be used for major operations where it would be unethical to do so, thus making it difficult for patient outcomes to be compared. In addition, standardization of factors such as the choice of anesthesia, pre-medication, and care after surgery would also be required in order to conduct clinical trials where patient outcomes may be compared, which is pretty difficult to do. Features and design aspects of clinical trials. Clinical trials require a large number of participants because people with the same disease can respond differently to treatments, and as such, clinical trials cannot be conducted with a small group of participants. One important aspect of clinical trials is that a comparison of efficacy is made between the therapy being tested and a control therapy of known efficacy. As such, a placebo or an active control may be used. A placebo may be defined as anything that passes as a therapeutic process used for treatment, such as a substance or procedure, but it has no direct biological activity. Meanwhile, an active control is an existing therapy of known efficacy. Clinical trials that use placebos may be referred to as superiority studies or placebo-controlled trials, while those that use active controls are often referred to as non-inferiority studies or equivalent studies. Superiority studies are considered to be more scientifically valid than non-inferiority studies. However, they are also considered to be less ethical. The findings of non-inferiority studies are considered to be less scientifically valid due to the fact that assumptions are being made about the efficacy of the active control based on previous clinical studies, some of which may have produced conflicting results. Thus, the efficacy of a new therapy can be difficult to accurately determine if the believed efficacy of the active control is actually incorrect. Meanwhile, placebo-controlled trials may be considered unethical where there are effective pre-existing therapies for severe diseases and medical conditions that can result in death or serious harm if left untreated. Thus, active controls are often used in such circumstances, because using a placebo would be unethical. However, if no permanent harm would ensue due to a delay in receipt of active treatment, then placebos may be used in a clinical trial. Clinical trials often utilize the process of randomization for assigning patients to each treatment arm, where each participant has an equal and random chance of being assigned to all three treatment arms. Randomization is employed because there are three possible reasons why there may be a difference in the health outcomes of patients in the different treatment arms of a clinical trial. They are the following. The new therapy being tested exhibits a genuine effect on the health outcomes of patients. The effect of the new therapy compared to the control therapy is simply due to chance. Or systematic differences, otherwise known as biases, exist between the different treatment arms and participants cannot be attributed to the different therapies received by patients. By assigning patients to different treatment arms randomly, randomization helps to ensure that each treatment group is as similar as possible, including helping to ensure that any risk factors are spread out reasonably evenly between different treatment groups, making the health outcomes of the two or more groups as comparable as possible. Consequently, the use of randomization allows for selection bias to be minimized, whilst also allowing researchers to determine as validly as possible the effectiveness of the treatment received by the experimental groups. Clinical trials that employ the randomization of patients into two or more treatment arms are known as randomized controlled trials, and are often referred to as the gold standard for evaluating the effectiveness of interventions, as they have a low risk of bias and reduce the influence of potential confounding factors. Clinical trials may also employ the technique of blinding, otherwise known as masking, in order to reduce the bias in the trial, especially with regards to exaggerating the efficacy of the therapy being tested. Blinding is consequently important for increasing the validity of the results produced in a clinical trial. Blinding is the practice of ensuring that certain people involved in a clinical trial are unaware of the therapy administered to each patient. People that may be blinded in a clinical trial include the participants, investigators, outcome assessors, care providers, data collectors, and data analysts, and any other staff involved in a clinical trial. Clinical trials may be single-blinded, where only participants or the investigators, but not both, are unaware of the therapy being administered to each patient. For example, a single-blind study examining the effects of nitrous oxide, which is laughing gas, was conducted where participants were deliberately not informed about the type of concentrations of the gas that they were breathing, so as to not introduce patient bias. Meanwhile, clinical trials where both the testers and the subjects are blinded, i.e. unaware of which therapy is being administered to each patient, are referred to as double-blind trials. For instance, consider a study testing the effectiveness of a new drug for a particular medical disorder where the experimental group receives the actual drug, whilst the control group receives a placebo. Within a double-blind study, both the participants and the researchers would be unaware of which participants are receiving the actual drug and which participants are receiving a placebo. Clinical trials that do not use blinding can develop bias, which may be generated by patients and/or researchers and clinical trial staff. Bias generated by patients includes A. Post-randomization selection bias, which is generated when a patient is unhappy with the treatment arm they have been assigned to, and so may be an uncompliant participant. B. Observer bias, where patients are aware of the treatment that they are receiving, act more or less positively to the treatment that they receive, depending upon their perception of the therapy. Meanwhile, bias that may be generated by researchers of clinical trial staff includes the following. A. Selection bias, where a knowledge of the treatment allocation to each patient may enable researchers to guess the next treatment allocation, which affects their ability to allocate future patients to different treatment arms without bias. B. Post-randomization selection bias, where researchers may exclude certain participants from the trial after randomized allocation. C. Observer bias, where researchers may record patient outcomes more positively for patients in a treatment arm to which they are predisposed towards. In addition, they may make repeat measurements when unexpected measurements are recorded, or their rounding of values up or down may be affected. It should be noted, however, that it is not always possible for patients or investigators to be blinded in a clinical trial, and instead blindness may be introduced when performing data analysis. Stages and phases of clinical trials. In Australia, clinical trials for medical and biological therapies often undergo phases of development, while medical devices often undergo stages of development. Recently, however, the structure of clinical trials has become more flexible, and thus do not necessarily fit well into the traditional different phases or stages of a clinical trial. The traditional different clinical trial phases of development for biological and medical therapeutics include Phase 0, used to gain a preliminary understanding of the action or actions of a drug. These clinical trials often involve a small number of participants, who are subjected to limited dosages of the drug. Phase 1. This is used to determine appropriate dosing levels, the most effective routes of administration, as well as a deeper understanding of the action or actions of the drug. This phase often involves between 10 and 100 participants, who may or may not be healthy volunteers. Phase 2. This is used to determine the clinical efficacy of a drug and its therapeutic dose range. This phase typically involves between 100 and 300 participants. Phase 3. where the therapeutic effect of the drug in the target patient population is assessed, and a risk-benefit balance is also assessed. Phase 3 trials often involve between 300 and 3,000 patients. Phase 4. Phase 4 clinical trials can be observational in nature, with safety monitoring of the drug after it has been released to the market, allowing for the safety of the drug in the general population to be confirmed, any long-term effects of the drug to be determined, and rare adverse reactions to be identified. Given that these trials can occur after market release, they can involve thousands of patients. Phase 4 clinical trials may also involve the assessment of the effects of the drug in conjunction with other existing therapies. Stages and phases of clinical trials. In Australia, clinical trials for medical and biological therapies often undergo phases of development, while medical devices often undergo stages of development. Recently, however, the structure of clinical trials has become more flexible and thus do not necessarily fit well into the traditional different phases or stages of a clinical trial. The traditional different clinical trial phases of development for biological and medical therapeutics include Phase 0, used to gain a preliminary understanding of the action or actions of a drug. These clinical trials often involve a small number of participants who are subjected to limited dosages of the drug. Phase 1. This is used to determine appropriate dosing levels, the most effective routes of administration, as well as a deeper understanding of the action or actions of the drug. This phase often involves between 10 and 100 participants, who may or may not be healthy volunteers. Phase 2. This is used to determine the clinical efficacy of a drug and its therapeutic dose range. This phase typically involves between 100 and 300 participants. Phase 3, where the therapeutic effect of the drug in the target patient population is assessed, and a risk-benefit balance is also assessed. Phase 3 trials often involve between 300 and 3,000 patients. Phase 4. Phase 4 clinical trials can be observational in nature, with safety monitoring of the drug after it has been released to the market, allowing for the safety of the drug in the general population to be confirmed, any long-term effects of the drug to be determined, and rare adverse reactions to be identified. Given that these trials can occur after market release, they can involve thousands of patients. Phase 4 clinical trials may also involve the assessment of the effects of the drug in conjunction with other existing therapies. Next, we have ethics in clinical trials. In the field of clinical trials, ethics and ethical regulations or guidelines investigate the welfare of participants by considering what is deemed right and wrong conduct. There are 4 key concepts, known as Beauchamp and Childress's 4 principles in clinical trial ethics. There's the principle of beneficence, where the delivery of benefits to the participants of a clinical trial are such that the risks therein are sufficiently outweighed. There's non-maleficence. This is the responsibility to prevent the harming of the participants in the clinical trial. There's autonomy, where the participants are able to make their own choices, have their own perceptions and beliefs, and are able to act on them. Then there's justice, where everyone is treated equally and the distribution of associated risks and benefits is as equal as possible. Informed consent is another important aspect of clinical trial ethics. Participants in a clinical trial need to be informed of all aspects of the trial, i.e. what will be done, what is being done for, and what their responsibilities and obligations are, what the risks and benefits are, or how long the trial will last. The participants of the trial must also be competent, meaning that they must satisfy the criteria of the study, but must also be competent enough to make their own decisions. Also, the participants need to be volunteers. In addition, informed consent is not an everlasting approval. It is a continuous concept that requires the participant to provide their consent periodically. The prevention of harm in clinical trials is also pertinent to the upholding of ethical integrity. Consequently, the use of placebo groups in randomized controlled trials can raise concerns because the administration of ineffective treatment to participants that require legitimate treatment can inflict harm on the participant. This can be mitigated by the administration of a dissimilar treatment that still demonstrates efficacy. However, there are concerns that the administration of different treatments to participants with the same disease is unethical. Clinical equipoise is another ethical concept in clinical trials and is where conflicting opinions on the benefits, risks, and other features of the various treatment arms of a clinical trial or study exist. Ethical considerations regarding these ethical trial designs are centered around the fact that no participant within any treatment arm should be subjected to a greater level of risk than what is presumed by the current standard of care. Hence, clinical equipoise ensures that all trial arms and placebos administer equal and adequate treatment with an efficacy no smaller than an available alternative. One of the key debates in ethics of clinical trials is the involvement of children and the potential harm a given trial may inflict upon them. In a paper by Simmons and Starkey in 2015, they explained that medical research on children is pertinent to improving their health and quality of life because research on adults cannot simply be expanded upon or modified to anticipate child health outcomes. This is quite simply because child physiology greatly differs from adult physiology, and their responses to given treatments and medicines differ. It can hence be viewed unethical not to conduct research and clinical trials on children. Risk in clinical trials need to take into account the age of the children being studied, risks, benefits, and possibilities of alternative treatments. The benefits of a trial may include the advancements of treatments, such as the advanced efficacy and safety. When considering risks, it's important to know that the risk can change over time due to advancements in knowledge, and hence risk must be continuously scrutinized and assessed. Agreement to be part of a clinical trial is predicated on sufficient knowledge of the study and thorough understanding. Children cannot give legal consent, meaning parents, legal guardians, or representatives must provide their consent, which is a continuous process occurring for the entire duration of the trial, and must be well documented. Simply not objecting to participating in a trial is not consent. Age is but one factor in the determination of whether a child is able to provide consent, but others include intelligence, stage of development, life experiences, and circumstances. Incentives in clinical trials involving children is another ethical dilemma. This is because incentives may cause participants to underestimate the risks of the study. What's more is that the incentives for parents, legal representatives, or guardians may cause them to underestimate the risks to their children. Ethical dilemmas in clinical trials involving children stem from practical difficulties. One example of this in pharmacokinetic studies is the taking of blood samples. This is considered an invasive procedure, and alternative methods must always be considered. However, the usefulness and alternative methods may not be as good as blood tests. For neonates, excessive blood sampling is considered unethical due to the small volume of blood that infants carry. For adolescents, ethical systems generally require the consent to be given by legal representatives. But this may change over the duration of the study depending on what constitutes the ability of the adolescent participant to make decisions. Confidentiality is also a key ethical concern in adolescent participants, most notably in studies concerning sexuality, violence, and drug use. The European Union Pediatric Regulation 2007 states that healthy children should not take part in clinical trials which may pose harm to them. In addition, in clinical trials involving various nations, comprehensive ethical assessments must be made in all of the countries that take part in the trial. Another ethical concern regarding clinical trials is the concept of post-trial access for patients who have participated in a clinical trial. Post-trial access is of ethical concern due to the fact that providing post-trial access to clinical trial participants, but not patients in the wider population until the drug has been approved for therapeutic use, creates a disparity between patients, which is of particular ethical concern for severe medical conditions. However, denial of post-trial access to the participants who greatly benefited from the test therapy and who have a severe medical condition and or whose health would greatly deteriorate if post-trial access is not provided may also be considered unethical. Now let's take a look at the disadvantages of clinical trials. Clinical trials require a large number of participants, such as several thousands, and as such, adequate recruitment of participants can be difficult, which can result in some proposed clinical trials not taking place due to an inadequate number of participants. This requirement for large numbers of participants has also led to the development of overseas trials, where clinical trials are often conducted by a drug company based in one country, for example, but the trial itself is conducted in an overseas population. However, this can create further problems regarding issues such as the notion of informed consent in different cultures with different philosophies, as well as different standards of care. For example, clinical trials in low and middle income countries can sometimes be the only way for afflicted people to access medical care, thus making these people vulnerable to exploitation. especially as there are health risks for patients who participate in clinical trials. In addition, post-trial access to the therapies tested is often another ethical issue, as many people in low and middle income countries cannot afford the therapies for several years after they are released, or the therapies may not even be available in their country. The requirement for large numbers of patients in clinical trials also makes clinical trials expensive to conduct, with the cost of a clinical trial increasing as the number of participants increases. This large cost of clinical trials has also meant that clinical trials are often funded by companies based in industrialized nations, and thus often test therapies for diseases that largely affect wealthy countries. In addition, clinical trials often take many years to conduct, resulting in an effect known as drug lag. This has meant that in some cases various therapies are at risk of becoming outdated before the results of the trial are published. This was particularly predominant during the AIDS crisis in the 1980s, where AIDS patients requested to access new drugs before trials for the drugs had concluded. In addition, for life-threatening diseases, drug lag can mean that affected patients can die before being able to access a life-saving drug. The placebo effect is where there is an improvement in the disease symptoms of a patient due to the administration of a placebo, or the therapeutic encounter from the administration of therapy. Consequently, the placebo effect can make valid comparisons of drug efficacy difficult. This is particularly problematic for a clinical trial if the placebo is quite large, as the larger placebo effect, the more difficult it is for researchers to assess the efficacy of a particular therapy. Large placebo effects can arise due to differences in the illness of participants, with some participants being more ill than others at the start of a clinical trial, affecting their responsiveness to the treatment and placebos, as healthier participants respond better to placebos relative to actual effective treatments. These differences in the illness of patients can arise due to variations in assessment of patient health by doctors involved in a clinical trial, and as such it is important when conducting clinical trials to ensure that patient assessment is standardized. Finally, we'll take a look at new developments and future directions. In adaptive clinical trials, researchers can change experimental design parameters, such as drug dosages and the number of arms in a clinical trial, based on data generated in the trial using Bayesian statistical methods. This allows researchers to make changes such as increasing drug dosages if low doses do not exhibit any effects, or to remove drugs with severe side effects, thus making clinical trials more efficient. This also then allows for unsuitable therapies to be removed from a clinical trial, rather than persisting with tests until the end of a trial. Consequently, an adaptive design for clinical trials can ensure that patients participating in a clinical trial also receive better treatment for their illness. Due to the high fail rates and expensive clinical trials, companies are also engaging in pre-competitive data sharing, where the results of failed clinical trials are shared amongst companies, thus preventing different companies from testing the same therapies already determined to be ineffective by another company. This ensures that research money is directed towards genuinely new, untested treatments, and thus may potentially increase the rate of development for new therapies. However, ethical considerations regarding the sharing of sensitive patient data must also be considered for such schemes. Collaboration between companies has also led to the development of platform clinical trials, where multiple therapies from different companies are tested for a particular disease during one clinical trial. Such clinical trials reduce costs and may increase the rate of therapy development because only one control group is required, rather than each company conducting their own clinical trial with their own control group. In addition, companies may also compare the efficacy of their treatment with that of another company during the trial. This allows companies to determine which therapy is best, and thus only the best therapies may be further researched, ensuring that money is not further wasted investigating therapies that will not be widely used by the public because there are better treatment options also available. Well done! You made it through the module. Now all that's left to do is the questions provided below, and if you get stuck, feel free to watch a video hint that'll direct you back to the section that it was covered. Good luck and thank you for watching.